Each cell has a nucleus, shown here as a purple-coloured structure. We normally talk about the nucleus during the interphase part of the cell cycle. That's when the nucleus is bound by a nuclear envelope or a nuclear membrane and not undergoing mitosis. The cutaway model of the nucleus you see here has a tiny circular structure coloured purple, which is the nucleolus. So don't confuse the large outer circular structure, that's the nucleus, with the little structure in the middle, which is the nucleolus. I'll talk about the nucleolus in a moment. Although not obvious in this model, the normal cytoplasmic nuclear ratio, the ratio between the cytoplasm and the nucleus, is usually about 3:1 or 2:1 in most cells. In cells that are immature or precursor cells, such as lymphocytes, that are yet to be told what really to do, then the cytoplasmic nuclear ratio can be about 1:1, which means they have very little cytoplasm. A lymphocyte, for instance, travelling around the body in the blood, is waiting to detect an antigen and really isn't doing anything. But when it does detect that antigen, it will then produce or grow its factory to create different components to help fight that antigen. And so the cytoplasm will increase in volume, but until then, the cytoplasmic nuclear ratio is quite small. Most cells that you will study in histology are mononucleate. In other words, they only have one nucleus. A couple of cells don't have any nucleus at all. For instance, the erythrocyte or red blood cell, or the thrombocyte or platelet. Some cells actually have two nuclei, or they're binucleate, and the skeletal muscle fibre can even be multinucleated. Have a look again at this image of a section of blood. Here's a neutrophil. That neutrophil has what we call a segmented nucleus. The nucleus is not a nice circular structure you normally would expect to see, but you can see it's divided into a number of little lobes. Look carefully at the nuclear membrane shown in the model here. You can see that it's a double envelope. We call it the nuclear envelope sometimes instead of the nuclear membrane. And what you can see here on the model is a nuclear pore. These nuclear pores occur all the way around the nuclear envelope. And they're very important because they allow the transfer of materials in and out of the nucleus. For instance, ribonucleic acid, or RNA, needs to travel from the nucleolus out through these pores into the cell cytoplasm. The nucleoplasm occupies the remainder of the nucleus, apart from the nucleolus, and includes a very fine scaffolding network of fibres within it and chromatin. Chromatin is represented by these dark and light purple-coloured regions and consists of strands of DNA and protein complexes. As I mentioned earlier, the most prominent structure you see in the nucleus is this large, circular, purple-coloured structure that you now know represents the nucleolus, which makes ribosomal RNA. Here is a histological section through the spinal cord and focusing on a large ventral horn motor neuron. The nucleus is this very clear circular structure in the middle. The nucleolus is a very tiny round dark circle you see in the middle of the nucleus. As I mentioned a moment ago, the nucleolus synthesizes ribosomal RNA. It's basically at least 85 to 90% protein, and the rest is RNA and DNA, all designed to synthesize ribosomal RNA, which is essential for carrying out protein synthesis. And again, that ribosomal RNA has to pass from the nucleolus out of the nucleus through the nuclear pores that I mentioned earlier. Here's an image from a section through the cerebellum in the brain, and the large dark brown stained structure you see is the cytoplasm of a Purkinje cell. A lighter brown circular element is the nucleus, and then this little round dot is the nucleolus. And that nucleolus, in those big cells, is the same size as the nuclei of some of these very small cells in the background. Those very small cells in the background are the granular cells of the granular layer of the cerebellum. They're the smallest cells in the body. They're around 4 to 5 micrometers in diameter. And so when you compare the size of those nuclei with the nucleolus in these big Purkinje cells you see that I pointed out earlier, you can see that the nucleolus is roughly going to be about 5 microns in diameter. Here's an electron micrograph image of the nucleus. It's a little bit distorted in shape, but it shows you the very dark stained nucleolus sitting in the nucleus, and all that dark stained material that you see within the nucleus is chromatin. Some of it around the outside of the nucleus appears quite clumped, whereas towards the centre of this nucleus, the chromatin is rather dispersed or lighter staining, and that depicts the two sorts of chromatin we have in nuclei. The lighter stained chromatin is called euchromatin. It's less compact. The darker stained chromatin is called heterochromatin. It's more tightly packed DNA. Now, when it's less compact, or when there's a lot of euchromatin inside the cell nucleus, it means that the chromatin is uncompacted and the DNA is unraveled. It's uncoiled. It's more open to transcriptional enzymes. It's more active, and therefore, by looking at cells, you can sometimes depict various activity of cells, particularly in relation to their production of proteins. Have a look at this image. Here, most of the nuclei you see are dark staining. They've got lots of heterochromatin in them, so the cells aren't that active. They're just sitting there waiting to be stimulated by perhaps a chemical messenger. It happens to be a section in the tonsil, where a lot of these cells are lymphocytes, just waiting to be programmed to do something. So they're essentially inactive, so their chromatin is very clumped. It's heterochromatin.